So like I said, I was going to take a recap. So from the chylomicron remnants, the liver was able to package some also, where the rough endoplasmic reticulum produced a special protein called B100. When it attaches to the packaged triglyceride and cholesterol ester, this particular molecule is called VLDL. When it comes here, when it's here, the HDL sees this. This is our VLDL. The HDL sees this and the high density lipoprotein and gives it two special protein, like I said, ApoE and ApoC2. When it does that, C2 binds to a particular receptor called lipoprotein lipase and breaks down triglyceride to free fatty acid and glycerol. The free fatty acid goes into the muscle where it makes acetyl-CoA, goes into the Krebs cycle and eventually finds itself in the ETC or electron transport chain and makes ATP, while some goes into the adipose tissue. The adipose tissue, and this is my muscle. The adipose tissue, where it reacts with the glycerol gotten from glucose, as I explained earlier, to form TAG. So after this happens, the VLDL gives back this particular protein back to HDL. Which protein? The same ApoC2. It gives back the protein back to HDL. Now what do we have? We now have this molecule. I have my triglyceride, low amount. I have some cholesterol and even some cholesterol ester. What is the protein that is associated with it? I have ApoE and B100. This particular molecule is called VLDL remnants, but scientists will prefer to call it IDL, intermediate density lipoprotein, or VLDL remnants. Isn't this cool? So you see, we are able to break down from fat globule to chylomicrons to very low density lipoprotein to intermediate density lipoprotein, otherwise called VLDL remnants. So once we have this IDL has an affinity for LDL receptors or low density lipoprotein receptors that are found on other peripheral tissues. It can also go back to the liver and do the same process, but the first thing it does, it binds to some LDL receptors that are found on some peripheral tissues. What is the first peripheral tissue? It's the adrenal cortex. So it can come here. So ApoE can come here and bind to this. So when it binds to this, so it binds to this LDL receptor. Yes, and it binds to this LDL receptor. When it binds to this, it shifts some cholesterol into the adrenal cortex. When it does so, this cholesterol converts this cholesterol to steroid hormones. So you know in the adrenal cortex you have zonal glomerulosa, you have zonal fasciculata, and zonal reticularis. So the zonal glomerulosa forms a steroid hormone called aldosterone. Zonal fasciculata forms cortisol, while zonal reticularis form DHEA, which stands for dehydroepialdosterone. So these are steroid hormones formed by the adrenal cortex. So the IDL binds to an LDL receptor takes in cholesterol. So it takes in cholesterol into the adrenal cortex, where zonal glomerulosa forms aldosterone, zonal fasciculata forms cortisol, while zonal reticularis forms DHEA or dihydroepidosterone. So when it does this, it can still go back to the liver. So now let's draw this diagram. Let's say it's coming back to the liver. So mind you, HDL can give it multiple copies. You can have multiple copies of ApoE. HDL is, it can, after sending back the C2, it can give it multiple copies of the HDL, the high density lipoprotein can give it multiple copies of this particular protein, ApoE. So some can bind to actually the LDL receptor that are found in the adrenal cortex. Others can come, but when it's coming back to the liver, now the IDL is coming back to the liver, it can bind with also the LDL receptor and continue to reduce this triacylglyceride, package the triacylglyceride, form more amino acid, save some cholesterol in form of cholesterol esters, and also form the cell membrane. Apart from that, there is a particular enzyme this IDL can bind to. So the protein B100 is able to bind to this particular protein. The protein is here. This protein is called, is called, sorry, mind you, this protein is called hepatic triglyceride lipase. Hepatic triglyceride lipase. So I told you IDL, after going to the adrenal cortex, it can also come back to liver, bind to LDL. The ApoE can bind to the LDL receptor related protein or LDL receptor. The B100 can bind to a particular enzyme called hepatic, which is found on the body of the liver, called hepatic triglyceride lipase, and still breaks down this particular molecule. When it does so, what do we now have? We have a particular molecule. So this is what will be remaining. After this happens, this is what is remaining.